Hi guys, in this video I'm going to go over the lab and hopefully clarify misconceptions. I may have mentioned that the lab as created in the lab manual is not 100% accurate or as it should be, and I did not catch this on time, so my apologies. I am going to go over the parts that are not at their best, and I would like you to make corrections in your lab before submitting it, as I have extended the due date for Monday at midnight. So again, I would like you to go back and make changes and make corrections onto your own lab. As all of you hopefully can agree, the experiment worked as it should have. All of us saw that the smallest cell was most efficient and was able to get the diffusion to happen the fastest. We had no problems there. However, in the section where you're asked to collect data, is where I'm having some difficulties understanding why you were asked to do that. So let's look at this data table. This is what you should have gotten in terms of numbers, okay? Surface area, volume, the ratio, diffusion depth, and diffusion rate, okay? I'm confident that these first three columns are completely fine in all of your labs. The diffusion depth, this is what was expected, okay? And I believe that most of you did get this. However, you are confused as to how or why it was the same value. Okay. If the numbers you got here did vary, that is okay. Okay, this is expected, and the diffusion can be affected by many factors, so it's not necessarily going to give us exactly what we expect. Okay. When it comes to the diffusion rate, that is accurate as well. And again, my issue is with these last two columns because if you were to graph them, you would not be able to get a conclusion based on a given graph. So I am proposing that we go differently or do one more step to actually find some kind of correlation between the cell size and the diffusion percentage that has occurred. Okay, when it comes to the diffusion depth, again, this column right over here, the measuring that you were supposed to do was indeed from the edge of the cube to the middle section, regardless of whether there is pink left or if there's no pink section left whatsoever. So again, the measuring should have been from the edge to the beginning of the pink section. Okay, and I believe that most of you did get the diffusion depth to be 5 millimeters per cube. Some of you may have had um, complete clarity or no pink section left in the smallest cell. However, you are still to only measure from this edge to this point, and that should still be around 5 millimeters. Again, if it was 4, 6, that's perfectly acceptable because that is how your lab specifically or your experiment worked. Okay. So instead of relying on these two columns right here and not being able to make a valid conclusion through a graph, we have a few options. Number one option is to find the diffusion percentage to find out how much of the cube was diffused into. That's one way to go about it. A different way to go about it that one of your classmates suggested is to, instead of looking at the diffusion depth, to allow the acid to diffuse completely and clear out the entire cube and to focus on recording the time. So to make the time the dependent variable, that is also a good way to go about it. I am going to ask you to stick to this option. Okay, so let me show you how you are to do this. You have the volume of the entire cube already given or found from your previous work. We're going to call that volume total volume. So TV is going to be my total volume of the entire cube. Okay, so I have 1 centimeter cube, 8 centimeter cube, and 27 centimeter cube. That is my total volume. I'm then going to find the volume of the pink section, okay, in each of the cubes. So that's my PV, volume of pink section. By doing that, I am going to then use that volume to subtract from the total volume to get the diffused volume. Okay, so again, PV is for pink volume, which you are finding by finding the volume of the pink sections that are left in here. To find the pink volume of each one, all you have to do is account for how much the diffusion depth was, okay, and then subtract that from the initial side. So if we're referring to cube number 1 with the side of 1 centimeter, after the diffusion, we are going to work with 5 millimeters less. Therefore, that is going to be 5 millimeters as well. So 1 minus 0.5 gives us 0.5 as well. So the new side, the side of this pink cube, is going to be 0.5. Okay, so then I will use my formula for the volume of a cube, which is V equals S cubed, S is the side, and I will find the volume of the pink cube within the cube. This is what I'm getting. So 0.5 cubed gives me 0.125. That is my pink volume. Once I have my pink volume, all I have to do to find my diffused volume, or this white area, this white volume, is to subtract the pink volume from the total volume, and this is what I'm getting. So the white portion, the volume of the white portion is 0.875. That's the part that was diffused. And last thing I have to do is find the diffusion percentage by simply dividing the diffused volume by the total volume and multiplying that with 100. So it looks like in cube number 1, the smallest cube, the diffusion percentage was 87% in my case with my data. Okay, yours could be higher, could be lower. That's a relatively high percentage, meaning that almost all of the cube was able to allow the acid to enter. The diffusion was really high. Okay, in cube number 2 and 3, I'm following the same steps. Let me explain them one more time. So again, I would like to compare the total volume, pink volume, and diffused volume. Total volume is the entire cube. Pink volume, volume of the cube. Diffused volume, volume of the white section that has been diffused. Okay, I already have my total volume from my table and from previous calculations. I will find my pink volume by simply using this formula. My side in this case is going to be 2 centimeters minus 0.5 centimeter. So I have 1.5 centimeters. Again, I have 2 centimeters minus 0.5. That gives me 1.5 centimeters for my side. I will use the formula. I will cube it and get the pink volume of this pink section to be 3.375. I will then find my diffuse volume by simply subtracting the total volume minus the pink volume, and I get this value right here. And last thing to do is to follow this formula. Or this one right here, I'm going to get the diffusion percentage by dividing the diffused volume with the total volume times 100. So I'm dividing the diffused volume of the white section with the total volume times 100, and I get 57% diffusion percentage, or diffusion in this case. So about half of the cube was able to, or the cell was able to complete the diffusion successfully. And then in the last cube, I'm doing the same exact procedure. It should be correct, and I'm getting 42% for the diffusion. Okay, now when you look at these numbers, it makes a lot more sense that the smallest cube would have the highest diffusion, the medium cube would have less of a diffusion, and the large cube would have the least diffusion, because as you guys know, small cells are most efficient because they are able to complete their tasks at a quicker rate and at a more efficient rate. Okay, once you have this done, the only thing that I wanted to address again is that your numbers could vary based on your diffusion depth. A lot of you had 555, but if you did not, that's perfectly okay. For those of you who put 10 for the 1 centimeter cube, again, you need to go back and simply understand that the measuring of the depth is from the edge of the cube to the center of the cube, okay? Or to the section where the pink dye starts. If your cube was completely clear, your depth is from the edge to the very center of the cube, okay? So it should be around 5. If it's not 5 exactly, that is perfectly okay. Last thing that I wanted to address was the graph. I am noticing that some of you are still struggling with the basics of a graph. I'm just going to quickly remind you as to what a graph needs to look like. You have two axes in a graph. You have the x-axis and the y-axis. Please don't forget this. The x-axis is where you're going to label and add your independent variable. Independent variable is what goes on the x-axis every single time. On the y-axis, we're working with dependent variable. Okay, so now at this point, you're to go back to your lab and figure out what was the independent and what was the dependent variable. Okay, you may have already answered this in the pre-lab, but let me try to help you a little bit. So the independent variable is that one thing that differed between the trials in your experiment. So we're talking about that one thing that was different in our case with the 3 cubes. What made them different? How were they different? That's what's going to serve as your independent variable. And then dependent, we were trying to see which cell is more efficient. And in our case, efficiency is represented by the diffusion or the percent of diffusion. If you graph the diffusion depth or diffusion rate, that technically is correct. But as we previously discussed, if you're going to, if you're going to rely on that data that was of the same numerical value, you will not get a decent graph. You will have a bar graph or a line graph with 3 bars of the same kind, and that will not give you any conclusion of any kind. Okay, so again, if you were to just graph using these numbers right here, you would get a single flat line if you were using a line graph, or you would get 3 bars with the same value, and that would just not give you any clarity or any conclusion. So you do want to graph the diffusion percentage. Okay, since we are technically comparing but also looking for a trend, you can use either a bar graph or a line graph. Okay, so again, x-axis, independent variable, kind of obvious what the independent variable is. In this case, what makes the 3 cubes different? And then dependent variable, in this case, the diffusion percentage, which directly correlates to the efficiency of the cell. Okay, hopefully this was helpful. If you have any questions, please let me know.